Hello and happy Tuesday. That's weird. I hope you all had a great Labor Day. Hopefully, you rested up, took a nice long break from your virtual learning. Hopefully, you did something fun. I did. Let's get back into it. It's been a couple of days, so let's kind of ease our way back in and refresh our memory of what we even talked about on Thursday and Friday. So, we started talking about sexual reproduction and how that all goes down. Today, we're going to talk about the actual phases and how that new combination of genetic information or your genes, how they are so different from the parents. So, you should already have your bell ringer done and you should have your guided notes pulled up. All right, what did we actually talk about Thursday and Friday before we dive right back in? So remember that sexual reproduction takes two parents and you have to have the sex cells or gametes from both, one of each, combine and their genes are going to combine to make that new offspring. So again, another term for sex cells is gametes. If you're a female, you have eggs. If you're a male, you have sperm. Now, how are the gametes made? How do you get those egg cells and sperm cells? So instead of mitosis, which makes the body cells, those gametes are made by meiosis. Meiosis is a lot like mitosis. Let me say that again. Meiosis is a lot like mitosis, except it goes through 2 rounds of cellular division instead of just 1. We separate these 2 rounds by just putting a number after them, meiosis 1 and meiosis 2. That's pretty easy. Also, meiosis results in 4 cells, and they're haploid cells, meaning they have half the amount of chromosomes. Also, last but not least, those cells are going to be genetically unique, meaning that their genes are not the same, they're not identical to the parent. You might also see it written as genetically diverse. Diverse just means different. So they are genetically different. Whew. Okay. Let's get into what these actual phases look like. Now, before we even start, you're probably thinking, oh my gosh, this is twice as long as mitosis, and mitosis was difficult enough. Fear not. They actually look a lot similar to the phases in mitosis. We even give them the same names because a lot of the same things are happening. So the first round, remember, is meiosis 1. During meiosis 1, those pairs of homologous chromosomes are going to separate into 2 new cells. Before we get into it, homologous chromosomes, just a reminder, homo means same. So these are chromosomes that pair up because they code for similar traits or proteins. So you're going to have your chromosomes for what determines your eye color. Those are going to pair up together. You have your chromosomes that maybe determine your height or your hair color or whatever. They're going to pair up together. If they are coding for the same trait, they are pairs. They are homologous chromosomes. So the first step in meiosis 1 is prophase 1. So the steps in meiosis are going to go in the same order as mitosis. So remember we talked about PMAT, the same thing for meiosis. Think PMAT. We're going to start with prophase, and in meiosis 1, it's prophase 1. So during prophase 1, those homologous chromosomes, each made of 2 chromatids, which remember is like half of the X, just like the one stick, they are going to pair up and crossing over occurs. Now, prophase 1 is hands down the most important step of meiosis, I would say. The most things happen in this phase. So if you get this one down, you're pretty much good to go. So what is crossing over? Here, let me move myself up a little bit. If you look in this middle, I'm pointing as if you can see where I'm pointing. If you look in this middle picture, you see those homologous chromosomes, and they literally cross over one another. Crossing over, easy. When they cross over, they actually swap some of their genes. Not all of them, just some of them here and there. So you can see that purple one takes a little bit of the blue, and the blue takes a little bit of the purple. This crossing over, and which makes a recombination, you might see recombination, which just means a new combination. It makes a new combination of genes, and that's why each new cell, after this whole process is over, is going to be genetically different. So because these chromosomes swap genes, That is why your sex cells are genetically different. They have different genes. So this is what happens during prophase 1. It's super important. So during crossing over, the homologous chromosomes, like I said, trade genetic information, and this is what causes that genetic variation. I still can't spell even after a long break. Okay, so moving on. Next phase. We got to remember PMAT, which means metaphase is next, and this is meiosis 1, meiosis 1. So we put a 1. So during metaphase 1, the chromosome pairs line up in the, remember M for middle, of the cell side by side. So they just line up as if they're like walking toward the arc 2 by 2 right in the middle. Easy. So we had prophase, metaphase, which means next is anaphase. And since we're still in meiosis 1, it is anaphase 1. So during anaphase 1, same thing that happened in mitosis with anaphase, they are going to separate and move to the opposite sides, or you might hear it called poles. That's the end of the cell. So this part's a little bit tricky. You remember in mitosis, the actual chromatids separate and split from that centromere, or the middle of the chromosome. Here, that's not quite happening. All that's happening is those partners, those two homologous chromosomes, they are no longer together. They're getting split up. They are breaking up. It's tragic. So they're just moving to the opposite side of the cell. Remember, A stands for away. They're moving away from each other. Okay. I keep moving myself all over the place. Last step, remember PMAT. So we have telophase 1, and then after telophase 1, we have that cytokinesis, which is when it actually splits. So remember in telophase, that nuclear membrane, or the nucleus, that new nucleus, will reform, and it's going to reform around that chromosome, which is now by itself. Remember, it doesn't have its pair because they separated. So that nucleus is going to form right around that and divide into 2 new cells during cytokinesis. Okay, so that is the end of meiosis 1. You now have 2 new cells, but remember, we don't stop there because we got to make 4. So next up we have meiosis 2. So what happens in meiosis 2 that's so different from meiosis 1? Honestly, there's not much of a difference except for 1 or 2 little things here and there. So one important thing before we even get into it is before mitosis and before meiosis, we always know that those chromosomes need to be copied so it can make those new cells, right? We don't copy again just because we're going through another round of cell division. We are still keeping the same amount of chromosomes that are in those 2 new cells after meiosis 1. So the chromosomes of the cell are not copied. Let's make this bigger. Perfect. The chromosomes of the cell are not copied before meiosis 2. They're only copied before meiosis 1. Okay. So what actually happens? Step 1 in meiosis 2 is, again, we're going through PMAT all over again, so it's going to be prophase 2. Easy. Now, here, again, because it's prophase, that nuclear membrane or that nucleus is going to break down, but this time you do not have crossing over. Crossing over, where those chromosomes swap the genetic information, that only happens during prophase 1. So the very first step that happens, and then never again. No crossing over. Next in PMAT is metaphase. So we have metaphase, phase 2, because we're in meiosis 2. Then the chromosomes line up in the middle, again, because M stands for middle. But now, since they're not paired up, now they are single file. So they are not 2 by 2 anymore, because they don't have a pair to even line up with. So they just stand alone in a line. Tragic. After M is A, so we have anaphase 2. Now here in anaphase 2, this is when you see those chromatids being separated. So that X is no longer an X, it splits up. This looks a lot more like the anaphase that you see in normal mitosis. So they separate and they move away, A for away, to opposite sides of the cell. Okay, one more phase. Whew. Telophase. We have telophase 2, and again, we have that cytokinesis. Okay, so now that you have all those chromatids separated, once again, that new nucleus is going to form around those new groups of chromosomes or chromatids, and that is eventually going to divide into 4 haploid cells. Okay, I just threw so much information at you, and I'm going to be honest, between you and I, this lesson is probably the most difficult and crazy one you are going to learn, honestly, the whole year. So if you're like, holy crap, Miss Long, like that was so much, we're going to practice it. I know it's a lot. Let's go back through it one more time quickly so we're not like got all this stuff jumbled up in our head. Okay, so with meiosis, it's like mitosis twice. So the first round, you have prophase, metaphase, anaphase, telophase, PMAT, like you would normally see, except in prophase 1, those homologous chromosomes, those chromosomes pair, swap some of their genes. Once that happens, they line up in the middle for metaphase. Then in anaphase, they are separated. And then in telophase, you have those 2 new cells being formed. Now those 2 new cells are going to divide again. So when that happens, you go through prophase, again, metaphase, again, anaphase, again, and then telophase again. So after those 2 cells split off again, you have 4 new haploid cells, meaning they have half the amount of chromosomes. They have half because you didn't duplicate your chromosomes again before meiosis 2 happened. If you have to watch that like 3 or 4 times to get everything that I'm saying, you're not, it's not you. Like it's a lot of information. I get it. Like I said, we're going to practice it. So let's do this one last slide here. This is the most important part of meiosis. This is why we learn it. This is what I really want you to take away from today. So because your cell gets only, because your cell gets only 1 chromosome from each pair of homologous chromosomes, the 4 new gametes, or the sex cells, are genetically different. What do I mean by that? If you look at this picture here, and I'm actually, I try to zoom in. I can't zoom in. I'm too old for this stuff. Okay, if you look at these cells going through meiosis, so once they cross over, both sides of that X, actually let me pull it back up here. Both sides of that X are not going to be identical. So like if you look at this first blue chromosome in this GIF right here, this side has some green, but the other side is fully blue. So both sides are not identical. Once they split off, this chromatid over here is going to have a certain set of genes, and this chromatid over here is going to have genes that are different. So that is why once you have your 4 new cells, their genes are not identical because they swap them out. This is why you, once you're born, may look different from your sibling, your brother or your sister, even though you have the same parents. It's because the sex cells that made you don't have the perfect identical genes to the sex cells that made your brother. Okay, so the genes in an offspring is a combination, let me get out of the way, a combination of chromosomes from 2 sex cells. Sorry, I'm looking at my stuff behind me. Okay, so we call this variation or differences. These differences in the chromosomes are what going to create the differences or genetic variation of each offspring. I'm sweating going through all that. Okay, If you have any questions about any of that, it's a lot of information. Hit me up. You know where I am. And we're going to practice it tomorrow and again and again and again. And eventually you're going to be like, Meiosis, I got it down. Easy peasy. Okay. Let me know if you need any help.